Institute, which is in Israel. I came on in August, and I'm staying here until this August, and then for another year. So there's plenty of time to collaborate and to discuss. Yes, part of the reason I'm staying another year is what's going on in Israel, which is a big disaster. But let's stick to science today. So what I will tell you today is a work, yeah. A presentable? What does it mean?
We can only see this and not the screen.
So
if you could.
Okay, so let me.
New share.
New share.
The screen.
Just want this. Okay. Okay. Does it work now? Okay. So it's a work that actually has its origins in a very long time ago, almost 20 years, when we were trying to figure out a way to describe theoretically a mechanism whereby cells can get their shapes. And now it doesn't work. Okay. And cell shapes in our bodies have a huge variety, as you can see here. And also they're very dynamic, as you can see in the movie, showing you an immune cell moving inside the three-dimensional gel. And it's clear that what's driving these shapes is what's called the cytoskeleton, which is a network of molecules that have been characterized and mostly built around these long biopolymers shown in this cartoon, which are made of this protein called actin. And the molecular components, as I said, have been mapped out, and they are numerous, to say the least, tens, if not hundreds of them, of course, all involved around the actin molecules, actin fibers, but then lots and lots of other molecules that control where does the actin polymerization into these long filaments actually occur, when is it turned on the membrane, and how is the actin filament, what's the network morphology of these actin filaments? Is it in a more branched network, or this, which is shown here, or is it this bundled network, which is shown here? So basically, the question that we are trying to do is how to bridge the gap between the molecular scale understanding, that people have a lot of understanding about what's the molecular components, and what is the resulting cellular shapes that the cells, that these components are supposed to be able to create spontaneously at the periphery of the cell in order to produce all kinds of shapes like you saw before, finger-like protrusions or protrusions that have more like the shapes of these sheets that are involved in many cellular functions like motility, engulfment of surrounding objects, etc. And we want to go beyond these cartoons, which are drawn in many papers, but are not the result of quantitative calculations. So coming from physics, that's the challenge of finding a mechanism for pattern formation, which we thought about could be based on a very simple mechanism that involves just three ingredients: the cell membrane, which is a flexible sheet, if you want to think about it this way, which is also a fluid, so things diffuse inside this sheet. Active forces due to the Okay, so sorry about that. Let me try to pick up from where we are. So The idea is really to couple these three components together to say maybe there is an interesting feedback mechanism that can lead to non-trivial shapes if what is guiding the cytoskeleton were to grow on the membrane is where these curved proteins aggregate. And since they aggregate these curved complexes, where the membrane best fits their intrinsic shape, like is shown here, where this one wants to go into this sharply curved region, maybe there can be an interesting feedback because those highly curved regions will be created by these pushing forces, which will now be localized to where these curved proteins are. So does this actually work? And can we get interesting, spontaneous self-organization of the membrane and the cytoskeleton just by coupling these? And the short answer is yes. And I'm just showing you these nice movies just to sort of celebrate because we just now published the last of these papers, which is the results of which are shown here, where over the past 2 years, we managed to use very successfully this coupling to show that it can lead to a very rich set of
have a box.
observed in cells. And
But
I'll just walk you through these results as much as possible today. So just to show that the basic ingredient of what we are talking about a protein that can be both membrane-bound, curved, and also recruit actin polymerization. This is just a name, an example of this RSP53, which is such a protein. If it's adsorbed in huge amounts on just a vesicle, you can see that it creates these bending of the vesicle of the membrane by itself because of its intrinsic shape. And in the cell, it is able to recruit also actin polymerization to produce these finger-like protrusions, which I hope we'll get to later on. So when this idea started off with me and AJ playing around with this coupling, as you see, a long time ago, by the age of this paper, we really only calculated linear stability limit, which means that we can work everything analytically, which is a lot of fun for a theorist, because then you can actually plot phase diagrams and you can calculate from analytic results as function of your model parameters in which regime there will be such a spontaneous symmetry breaking and pattern formation, and under which conditions it will not, and everything will stay uniform, which means that any small perturbation of the uniform state will go back to being uniform. So for example, what's shown here in this scheme is that if you have, for example, a higher concentration of these proteins here and here and lack of them in the middle, then in the next time point, which is denoted by this arrow, there will be a larger deformation here because the force that each of them is producing is larger here because it's like a center where actin is nucleating and pushing. And since they're also curved, these objects, by this strange cross-section, they like this high curvature region, so they further aggregate there, as denoted by these small arrows, because they can move around in this fluid membrane. And therefore, you see that the initial perturbation, which was almost uniform, there was a small gap here, now became even larger. And that's a positive feedback that can lead to runaway instability. The only problem is that when you do this linear stability analysis calculations, you're having a lot of fun because everything is analytical, but you're limited to just small perturbations. So you don't know what will really be the end result of this positive feedback. What kind of membrane shapes will really be created when this thing can run its course all the way? And all this work, which is mostly linear stability analysis, is summarized in this review paper, which is also a little bit old by now. But then, right after it came out, I started a very nice collaboration with this group of very nice people in the University of Ljubljana in Slovenia. And the work, which I will show you, was mostly, almost all of it done by Raju, who was a postdoc in my group and now in Curie, and Alesh and Veronica and their group members. And again, most of this work some of it is summarized in this recent review article. And what allowed us to go beyond the linear stability analysis is numerical simulations. There's no other way around it. And it's based around having a vesicle, or basically a membrane sheet, which is closed, so it has the topology of a sphere. And if you zoom in on it, it's basically a triangular network. on this network, you can give each node some property. For example, you can give it a property that it has 0 spontaneous curvature, so it wants to be flat, or we can give it a finite spontaneous curvature, so it wants to bud out. Like you see with these red nodes here, which bud out, you can give it an opposite curvature, so they want to go inside. And basically, you're doing a Monte Carlo calculation. A Monte Carlo calculation means that you're adjusting the position of each of these nodes at some random direction, and you accept such a move if it lowers the total energy of the system. And if it doesn't lower the total energy of the system, you can still accept it with some Boltzmann factor, which means that you allow thermal fluctuations to kick you up in energy. And what is your energy that you are calculating? The simplest one you can imagine. It has the smallest number of terms you can imagine. One is the bending energy. So to bend a bilayer costs energy. So basically, we assume that we are only considering the mean curvature, which is denoted by h in each position I. And we are only looking at the mean curvature because all I will show you today deals with such curved objects which are completely isotropic. So as you see here, they cause a completely a uniform bending. So they're like small caps. So if you bring many of them together, they form a sphere. They have no directionality in the plane of the membrane. And that's actually not what's in cells. In cells, like this IRS-P53 curved protein that I showed you before, is actually banana shaped, which means it's highly curved in one direction, but 0 curvature in the other direction. So that's called anisotropic curvature, and that does not couple only to the mean curvature. But for now, all I will show you, and surprisingly that's rich enough already, is limited to such isotropic shapes. So bending energy basically means that you want to minimize this mean curvature because it costs you some energy to have a non-zero mean curvature, unless you have some spontaneous curvature denoted by C0 at some site, and then basically the membrane wants to conform to the shape that you endowed this protein. Now, this protein, this red node, can diffuse around. How can it diffuse around? Because we are doing T1 processes where we are taking one of these nodes, and instead of connecting these two, one of these edges, which instead of connecting these two nodes, we change to connection along this line. So by doing that, we are effectively fluidizing the system. So bending energy, that's the minimal energy. What other energy? We allow these two proteins, if they are close enough, nearest neighbors, to have some energy of binding, minus W, which we completely fix in all I will show you. We don't play with this parameter. And we also don't play with the bending modulus, which is usually a few tens of kT. And we keep this W to be small, around 1 or 2 kT. If it's too large, then they make aggregates which are solid-like, and they don't undergo detachments and reorganization. And we want to keep the system dynamic so it can explore the minimum energy configurations. Where's the cytoskeleton? So far, this system is completely in thermodynamic equilibrium. So we put in the cytoskeleton effectively. We have no way at the moment in this model to describe all this structure of actin polymerizing, branching, capping, depolymerizing, etc. So we simply say that wherever we have such a curved protein, there is a pushing force or a pulling force. It's equal, the same way they want to think about it, which takes this node and wants to pull it or push it from the inside in the direction of the outwards normal. So you see that the outwards normal here is not the same one as here, but the force is always the same, F. And it's treated as an energy term. F times the distance that you've moved this node along this normal is energy. Force times distance is energy. So that's, all the energies, of course, are normalized by kT. This is a non-equilibrium term because it's unlimited from below, so it's like an infinite external field. So far, that's only the 3 energy terms that are intrinsic to the membrane. There's one extra energy term that we add if we want this object of yours to interact with something external. For example, to adhere to some external surface. If you want that, you have to define some adhesion energy, which again, we define so that every node of this surface, which is just close enough means the length of the edge of our triangular network, close enough to this rigid surface. All the surfaces that I will show you are rigid. You can say that's not always true. Cells interact with soft surfaces that will be explored in the future. And you say that each of these nodes has some minus negative adhesion energy EAD, which is a constant. How does a simulation like that look if you start with this strange bipyramid shape, which obviously is not the minimum configuration for minimizing bending energy because of these sharp edges. It just minimizes the energy, and you see that it continuously fluctuates with these proteins moving around. There was one that was on its own, and then it joined this aggregate and can split from an aggregate, etc. That's, of course, highly coarse with a very small number of nodes, just to illustrate what does it mean to run such a Monte Carlo simulation. So just to remind you, the Monte Carlo simulation doesn't take into account the fluid dynamics inside this vesicle and outside. We don't calculate that. So what are we actually calculating? What are these movies? These movies show you in shape space of this object which way the system would want to go down the energy gradient, okay? So you are sampling correctly the thermodynamic or the energetics of shape spaces. You don't have a real clock, because you don't calculate correctly how quickly it can change this shape by pushing around the fluid around it and swishing the fluid inside it to fill up the new shape, right? All of these fluid dynamic processes in a viscous fluid, they are dissipative, and that's exactly what's controlling the speed. The vesicle moving in honey will move much more slowly than in water, right? So all of this time scale we don't have. So we have Monte Carlo steps, but it's not a real clock. So just to remind you. Okay, but besides all of these limitations, it's still useful. I hope I will convince you. So let's start with a passive system. You let these proteins, which are highly curved, stick to each other. You remember there's this minus W interaction. And not surprisingly, they all want to stick into one very long aggregate that has this shape of many, many beads stuck to each other. That's a theoretical calculation, analytical calculation. And that's not surprising. It might take a very long time to aggregate all of these in one single aggregate, but you see where it's going. There's a small energy barrier here, which has to do with this energy, this bending energy of the bare membrane between neighboring aggregates that prevents them from easily actually merging together. Thermal fluctuations can get you over this barrier, but it just takes a very long time. But you see where it's going. So far, so good, nothing very surprising. But what happens now when you turn on the active forces? So remember that the active forces, they act by the rule that we chose. Nobody here objected, that they are just pushing towards the local outwards normal. So what will happen to a structure like this? The force here will be pointing to the right, and here it will be pointing to the left, and so on, all around this tube-like object, and will basically inflate it. As soon as it will inflate it, the radius of curvature will not be anymore the perfect radius of curvature which fits the intrinsic radius of curvature of these objects. So they will be, from the bending energy point of view, frustrated. So what would be the next best thing that the system can do? So again, with other simulations, we could not guess, but only afterwards you say, aha, clearly this is what the system wants, because now all the forces are radially pointing outwards. So although the volume is not conserved in these calculations and is of course much reduced because it's now a pancake with very little volume in between, but now all the proteins are enjoying the very high curvature, at least in one direction. In the other direction, they're still frustrated because this radius of curvature is much, much larger than what they want to make. You remember, they want to make these small balls, but it's actually very, very stable. You don't see any protein just meandering here along this region. And already that sort of showed us, aha, maybe just by coupling these curved proteins with a protrusive force, we can get an explanation for why the cells have a very strong tendency to create these sheet-like protrusions, especially when migrating. But when migrating, they migrate on a substrate. So let's put everything on a substrate and everything will become more interesting. So I promise you. So if you put a vesicle which is completely empty and devoid of this protein on a very weakly adhesive substrate, it doesn't even spread. You can actually convince it to spread simply by loading it with lots of these curved proteins, still completely passive. The force is zero, there's no pushing force. This is in thermal equilibrium. And the reason is that these highly curved proteins basically remove this high energy cost of holding a sharp rim. So now the system can enjoy both the adhesion energy and still not pay the high energy cost of having a sharp rim. if you remember, this is only when you are giving this vesicle a surface. When the same system, identical system, was in floating in space, they actually much prefer to make these shapes. So this is only brought about by the fact that they are in contact with an adhesive surface. Now let's turn on the forces. So first I convince you that the passive system, if you simply don't have many of these proteins, this is 10%, before I showed you 17%, it's not very effective anymore in spreading. Now you can actually get a very robust spreading with the same 10% of these proteins simply by turning on the force. You see, for the first time, I'm showing you now with the force. This is very similar to what happens when a cell spreads. Many cells spread completely like a pancake with lots of actin polymerization along the rim. The rim is super sharp. And of course, we miss this big bulky nucleus in the middle. But you can say, okay, you've shown me a pancake floating in space. Why should I be excited by a pancake adhere to a substrate? It makes even more sense because you gain the adhesion energy. So wait, I hope you'll see something nice in a second. First of all, what happens if you simply don't have enough of these proteins to make a full ring around the vesicle, the spread out vesicle in the pancake shape? The system spontaneously makes the best next thing that it can, which means local spread out arcs around each of these edges. But since you don't have enough of them to link them into a full ring, and there's a bit of a gap between them, the net force of one is to the right and the net force of the other one is to the left. And basically they stretch whatever's in between them, which is just the bare membrane. Okay, and some cells look actually highly spread out like a spindle when they are given an adhesive surface, but no motility. So what we found is that, in fact, if we are in the same regime where we don't make a full pancake, but we can make this two-arc shape that I just showed you before, but we actually reduce the force and crank up the adhesion energy, we can get into this triangular shape region where the system spontaneously forms a single arc. So it doesn't break, it can break into two arcs, but as I'll show you in a second, it can coexist in this single arc. And since now this single arc has a net force in one direction, lo and behold, the object begins to move. It's alive. Well, I don't know if it's alive, but it's moving like a live object because it resembles a lot motile cells which have a front full of actin polymerization, which is exactly the place where actin is pushing in our spontaneously forming object or mini cell or whatever you want to call it. It has also the overall geometry of a moving cell, so a very sharp front and a very fat back where basically, well, I shouldn't be insulting, but the back is basically trying just to minimize the bending energy. Of course, again, we are missing the bulky nucleus. Just to show you that this already can be interesting to explain some observations. So we came across this paper by Rob Insall and Laura, where they actually interfered with the normal cell ability to nucleate actin. And the wild type cell actually has these two arc shapes very often, maybe even three arc shapes. So not very polarized and not moving very much. But actually by down-regulating some of the proteins that polymerize actin, it switched into this highly motile crescent shape. So maybe we can rationalize this by actually explaining that indeed, if in our model, the force of actin polymerization is too high, you turned it off again. Yeah, is too high, you are stuck in this two-arc shape, and actually tuning it down gives you this this crescent shape, which is much more motile. What happened? Okay, but the system is actually very fragile. And as you can see, if there is a chance fluctuation that can break this leading edge cluster, it breaks spontaneously into this two arc, which cannot continue to move anymore because it's almost not perfectly balanced between the arc on one side and the other. And this actually, in fact, is observed in experiments. For example, another experiment by Rob Insall. This is a cell moving to the right with a big leading edge. Then spontaneously, the leading edge breaks into something that looks like a two-arc, but real cells have a mechanism for repolarizing and continuing along a new direction, as you can see also in the following movie. So you see that it had a nice single leading edge. It broke into two, but the cell at the end of the movie resolves this competition, and only one of them wins, and the cell continues to walk roughly at 90 degrees. We don't have that yet implemented in this simulation, although we are working on it, and it's already working, but it's beyond this talk. Furthermore, if you run this object against a rigid wall, because the highly curved leading edge becomes squashed against the wall, so its high curvature is completely lost, and these proteins like high curvature, they immediately go to both ends, on the right and on the left, and again, you're ending up with this cell object which is not polar. Cells do lose polarity when they hit barriers, but again, they can recover polarity and move off. Our object cannot. So what can we do with it? We can actually explore it on very smooth surfaces. Just be very gentle to this object, which has come alive. And this is a paper which came out just 2 months ago, I think, 3 months ago. Yeah. And I'll show you the results. So we came across this paper from a group in South Korea, where they looked at cells moving in a sinusoidal surface. So we said, sinusoidal surface is interesting. Let's look at sinusoidal surface. Maybe our object will survive a sinusoidal surface. And you see what they found. They found that when the wavelength is such that the cell can snugly fit within the groove, it actually prefers just to move along the grooves. These are those straight lines in these trajectories. And when it goes over a ridge, it almost moves at 90 degrees, as if to minimize the motion over the ridge as much as possible, and then settle into the new groove. So let's see what our model does. We thought that maybe there is a chance that our model will work to explain this behavior, which is called curvotaxis. There's chemotaxis, which you've heard about. Curvotaxis is how cells respond to a curvature of a surface. And why? Because we thought, we have curvature sensitive proteins which create this leading edge. So they are curvature sensitive to begin with. So that must give our object some curvature sensitivity. Maybe it'll work. It's wrong. It's wrong thinking. It works, but for the wrong reason, for a different reason. So first of all, let me show you that it works. We can start with our object on the ridge, and it spontaneously slides down. Of course, there's no down and up. There's no gravity here, but it looks like it's sliding down when you look at the movie. When you start it aligned along the pattern in both cases, but now inside the groove, it just meanders in the groove, but stays in the groove. So this looks very much like the observed migration pattern. But what's interesting is that if you simply change the wavelength of that sinusoidal pattern such that this object of ours, whatever you want to call it, this mini cell, doesn't fit snugly within the groove, but is now, because of the wavelength, which is smaller compared to the size of the object, it spills over to the opposite curvature, right? Because the groove has one sign of curvature and the ridge has another sign of curvature. So if the cell is obliged by size to always sample also the other curvature, it now prefers to switch completely and move actually perpendicular to the pattern spontaneously. Even if you start it along the pattern, it will rotate to move along the pattern with the velocity which is accelerating up and down. And that was actually seen by experiments on keratocytes in Sylvain Gabriel's group in Mons. And again, the same model does both, moving in the groove when you're small, like here, and moving across the grooves when you are larger than the pattern. And the question is, of course, why? Now, since we have all the energy terms and all the system is trying to do is just to minimize energy, we can interrogate, quote unquote, the results and see exactly which energy term is driving this behavior. The only problem with really understanding it nicely is that when you have an object which is constantly moving over different curvatures, it's difficult to understand still, although you have all the numbers in front of you. So we thought it'll be easier to understand when we put our motile object, our mini cell, on a surface with a single curvature, like a fiber. And here we found that if you start it along the length of the axis of this fiber, it spontaneously switches to rotation. And now we can ask why and maybe understand better. And first of all, this immediately tells us that basically by going over the ridges, it's doing exactly what it's doing here. It thinks that it's trying to rotate over something which is curved like a fiber. That's what it's doing over the ridges, and that's why the ridges naturally align it to be perpendicular to them. And the reason it's doing that is actually very subtle. It's not because of the curvature energy of the individual proteins along the leading edge, because their spontaneous curvature is super high. That's why they maintain this very sharp front by aggregating there and pushing with the active forces in order to continue to maintain this sharp front. But this sharp front is a very high curvature. So the curvature changes of the surface are minuscule compared to that. So it's not their direct curvature sensitivity, which actually gives rise to this global emergent curvature sensitivity or curvotaxis. What happens is that the global adhesion energy and slightly also the bending energy is reduced as the object changes from axial to circumferential. And we think the reason is that if it's along the axis, all the forces which are pushing along the axis are simply moving the object forward and don't change the shape. The only way that the shape can change is from those forces which are on the flanks and can actually increase the coverage of the surface, which is the adhesion energy. But when they are aligned like that, it costs a bending energy to do that. When you're aligned along the circumference, again, the pushing force, which is along the which is circumferential, simply moves you along. But now the flanks, the sides, are spreading you along the minimum curvature direction of the fiber, which has zero curvature along the axis, and therefore they are more effective in spreading. And the excess area is exactly the additional adhesion energy and lower bending energy. So it's a subtle emergent effect. It has not directly from the energy of the binding energy, of the bending energy, sorry, of the individual proteins, although they are curvature sensitive themselves at the leading edge. So we looked for experimental evidence, and in fact, it languished for 7 years in a PhD thesis. Just to show you'd never throw away a PhD thesis from the group of Eberhard Bodenschatz in Göttingen. And they found that when they put dictyostelium on a fiber and they look at it from the side, they can measure the directionality of the motion, and there's a clear peak that cells prefer to move in this direction. They can't see a whole rotation because of the optics. Once the cell goes over the fiber, the optics through the fiber is, of course, a big mess. And there was another nice result from Carsten Beta, where now he let the same dictyostelium move on a surface and then climb up a pillar. So now when you look down the pillar from above, you can actually see full rotations. Here it doesn't actually make a full rotation. It's also throwing out some protrusion to touch the other pillar, which of course only messes things up. But in general, we have nice chemographs of them going on persistently, and they do tend to want to rotate, of course, equally right or left. What happens now if you go back to our shapes? You remember that we could get a motile object with a single arc of these curved proteins, but it coexists with this two-arc object, which is not motile. So what happens when we take this object and we put it on a fiber? So maybe you can already guess, because each of these ends is like, by itself is like a leading edge. So you remember that the leading edge by itself wants to become circumferential and point circumferentially, even though you started off axially. So if they would both go circumferentially in the same direction, they'll just merge and you'll end up with this giant object rotating. But if they merge, if they turn circumferentially in the opposite directions, what will happen? Oops, why is it like this? Ooh. Okay. Ah, now it's much nicer. So it's, yeah, it's whatever you want to call it, a boa snake trying to strangle the fiber, trying to coil, but runs out of membrane, so they're brought together at some point. We don't allow them to cross into each other. We don't allow them at the moment also to adhere to each other, so they just don't penetrate. And we found nice evidence for this in how cells send out protrusions along fibers from the work of Amrinder Nain in Virginia Tech, where he places cells on these artificial fibers of very small radii, highly controlled. And you could see that the front of these protrusions, as they are being sent outwards, is continuously rotating in a similar spontaneous manner. Now, since we have a calculation that we can calculate all the behavior, we can actually map out the different phases of what does in principle our vesicle want to do when presented with a fiber. It's not always coiling. If the radius is too thin, it cannot actually make coiling. And of course, if there's too many of these proteins, instead of making these two arcs, they just make the pancake, and the pancake can either coil or not coil depending on the radius. So that's a very simple phase diagram. Oops, sorry. And we could actually make a prediction that this coiling will cease if you actually make the cross-section shape of this fiber such that it has such a sharp edges by making it more and more oval until at some point the bending energy cost of coiling is too much and the object will only elongate along this axis. And they found a similar behavior by taking their 200 nanometer fibers and basically pressing them down into a flat ribbon, which I agree is quite extreme, but at least it goes in the same direction. Where can you see coiling in a more biologically sensible setup? So I was told to look for myelination of axons. I don't know why this is flashing like that. So Ori Peles's lab in Weizmann was doing such work, where basically in red you see axon of pre-plated neurons in the Petri dish. And you see here that what is called the Schwann cell, never heard about it before, but maybe you've heard about it, is a cell that does myelination. And what's actually interesting is that this is happening days before the real myelination happens. So when we went into the lab and asked them if they see such coiling of protrusions, they said, the Yael, who was the girl who's doing these experiments, she said, I've seen it, but we've never reported it in any papers because this is such an initial stage before there is the actual coiling of the myelinated membranes that they were never interested in that, because that like happens days before there's the full-fledged myelination. But I think it's quite nice pictures. So I've shown you fibers. Why does it do that? Okay, I don't know why. Can it run a movie without doing that? No. Okay. This is terrible colors. Okay, so let's jump over this phagocytosis behavior because it doesn't want to run the movies nicely. Okay, maybe here. Why not? Okay. Yeah, it doesn't play. Okay. So maybe I'll go into the next, maybe one of the last parts. Okay, I hope here the movies work because they are nice, I think. Okay, so I've shown you that by coupling curved proteins which are protruding outwards with a pushing force, you get spontaneously sheet-like objects. But we know that cells also make finger-like protrusions. So how can our model make them? So one way to make such elongated protrusions instead of pancakes is to actually reduce the spontaneous curvature. So to make these proteins which are highly curved less and less curved. So in fact, if they are completely flat, they will make such protrusions, but then it predicts that this cluster of proteins which are nucleating the actin at the front of such finger-like protrusions has to be actually very flat. And the problem with that is that the same IRSP53, which is indeed highly curved and found in the leading edge of flat sheet-like protrusions, which are usually called lamellipodia, is also associated with these finger-like protrusions called philopodia. For example, these images from the lab of Shiro in Nara, Japan. So what are we missing? So what you have, whose picture is there, who used to be a master student, now he's left science for much better paying Intel. He's, yeah, unfortunately, the big changes that you see in these finger-like protrusions, the actin has a completely different architecture than in these sheet-like protrusions, where the actin is branched, and therefore you can think of our rule that it's like just a local isotropic pressure. So wherever these proteins are, it's just a local force which is towards the local outwards normal. Here, it's clear that all the actin is bundled. So we wanted to implement it by simply borrowing on an idea that comes from the physics of flocking, of how to describe how a flock of starling are moving through the air just before sunset. And that is to say that the local force applied by each of the proteins inside this single cluster is affected by its neighbors, so that it tries to align with its neighbors. And you see that this group of two are in a disconnected cluster, so they are aligning with themselves. So if you apply that, and that would be our poor man's way of describing this bundling of actin without actually going into description of all these bundling proteins, etc., lo and behold, we can get, if the radius of this alignment and the strength of this alignment is strong enough, such elongated structures which look like these finger-like protrusions, like philopodia. They always get this bulbous head in our picture, in our model, and this sleeve-like structure. If you look at the movie, I hope the movie works. Let's see. No, not really. Okay. Okay, I hope this was actually a nice movie. Okay, so now that we have proteins that can make sheet-like protrusions and proteins that can make finger-like protrusions, what happens if we mix them together? So the red here are those that just want to make the pancake and this flat sheet-like protrusion along the edge, and the yellow are equally curved, so they have the same spontaneous curvature, but only the yellow with neighboring yellows want to align. And here, this simulation, I don't know if it works or not. Let's see, probably the colors will go crazy. Oh, it just doesn't work. Okay. Here we assume that they mix completely with each other. So these are snapshots from this movie that I couldn't show you. And what you see is that they are binding, red and yellow bind to each other equally. Still, the yellow can aggregate enough of them into the tip of this structure, which is still like a philopodia, if you want, although it is highly decorated by red along the length of the bottom part. And all along the edge here, you have red and yellow mixed together. Remember, the yellow along the edge is frustrated because yellow alone only wants to make fingers. It doesn't want to make these sheet-like protrusions. And in the bottom, you see a similar movie where now we assume that yellow only bind to yellow and red only bind to red. What's interesting here is that although the yellow and red don't bind to each other, you still have, this long protrusion, this philopodia-like structure, just like here. Here it's actually more segregated to all yellow in front and stronger red in the base. But this red in the base of this finger-like protrusion is frustrated. You remember, the red alone wants to make sheet-like protrusion, doesn't want to make cylinders. But what's more interesting is that along the length along the edge of the rim, you have interspersed yellow, red, and the process of yellow finding more yellow and then able to amass enough of them to make a new protrusion is actually blocked, because the only way this yellow can merge with the yellow here is to get rid of this red. But this red is highly curved proteins. They don't want to go into this flat region, neither do the yellows. So actually you are reduced to a one-dimensional system where coarsening is actually blocked by these traffic jams. So each one of these domains is blocking the further coarsening of red to red and yellow to yellow. And that's actually cute, because first of all, if you look at the philopodia, although the philopodia has bundled actin, which is very different from the branched actin, you still see a lot of nucleators of the branched actin penetrate the length, especially the bottom of such a philopodia. That's a work from Laura Maczewski from quite a time ago. And there's other papers that show similar behavior. So the fact that we get red to infiltrate the bottom of this philopodia can be supported by observations. And second of all, as you see here, along the length of the front of this lamellipodia, which is this sheet-like protrusion, you get lots and lots of these what's called microspikes. And these microspikes are exactly bundled actin, which is very different from the branched actin in between. It's bundled and it's actually nucleated by another nucleator, which is different from the nuclear in between these microspikes. So again, you get exactly the same kind of interspersed organization. And this paper comes from the group of Clemens Rotner and Jan Fikes. And when I met Clemens last week in Birmingham, he was actually telling me that it still puzzles him. Why do the nucleators of these microspikes, which alone by themselves produce very nice philopodia, when you block the red proteins, when you block completely the ability of this cell to make the sheet-like protrusions, when there is the sheet-like protrusion, they're also happy to be interspersed inside these sheet-like protrusions, but never reach a critical amount to create a full-fledged philopodia that comes out. And I think this just suggests that there is nothing to it beyond curvature sensitivity, that the yellow has the same curvature sensitivity, same spontaneous curvature as the red. What's driving this sheet-like organization is the red, but the yellow is just stuck there and never is able to coarsen enough to form a fully fledged philopodia or finger, which is really what it wants to make if you give it some intention. Now, the last thing I want to show you is what happens when you mix two types of curvatures, because all I showed you so far in all the works, even though you have here two types of force production, normal force production or aligned force production, the spontaneous curvature of all of these proteins was curved outwards. What happens if you mix proteins which are some curved outwards and some curved inwards? I hope this, no, it's a complete mess. Okay. Okay, so let's just look at the still pictures. Okay, so what happens is the following. What we chose is that those that are curved inwards, which are in blue, are passive. They don't produce any forces, but those that are in red are the same as before. They are curved outwards and they can also push outwards. So first of all, you can say what happens if you mix the two of them and both are passive. Yes, you get similar behaviors. The red like to line the edges or shoulders of this tunnel that goes into where you have this invagination of the blue into the vesicle. But the hole is actually rather big. You can see the hole here with your eyes. If you let the red also produce pushing forces, then you can get that, first of all, the segregation between them is much stronger. There are less events of red being stuck like this Spider-Man shape inside the blue. And the hole tends almost to squeeze to zero. In fact, it wants to squeeze to zero, but you see it's the minimal size that our calculation allows us, which is a single such triangle. If we would allow a fusion, that's where a fusion would actually be driven, and this whole vesicle will be just cut and thrown into the vesicle, just like endocytosis. So is it like a very primitive model for micropinocytosis or actin-dependent endocytosis? I can just show you, I think, nice pictures that come from the group of Rob Kay and Till Bertschneider. Till is from Warwick and Rob is from here, from Cambridge. And they looked at micropinocytosis. And again, superficially, the shapes look similar. So, maybe it could say something about how actin is spontaneously organizing around the lips of such closing patches that eventually endocytose or produce a vesicle into the cell. I think I have to, okay, so I won't have time to talk about shear flow. If somebody's interested, you can talk to me afterwards. And I hope in the spirit of the name of this seminar series, I hope I convinced you that physics can maybe help us bridge the gap in a quantitative way, not just drawing cartoons that we think is what happens, but by really calculating shapes that are determined by spontaneous curvature and forces we can have very rich mechanisms for spontaneous formation of all these rich behaviors that we see that the cell is doing, like phagocytosis, which I couldn't show you, to cell migration, curvotaxis, etc. In the future, we want to take into account anisotropic proteins. We already have it in the code, but we haven't actually started doing that yet. So proteins that are really banana-shaped or even saddle-shaped, like in nature, contractile forces, which are very, very much there, and interacting vesicles. So even the vesicle can interact with itself, like the coiling, and maybe then we can have something which is multi-layered, more like the real myelination process, which is very interesting for people studying the neurons and axons, and also how two of these objects interact with each other, like two cells interacting with each other. I don't know if it's one universal mechanism for actin-based cell shape dynamics, because the cells are surely also using reaction diffusion mechanisms for pattern formation. So it's not only based on this particular coupling between shape and force, but also reaction diffusion and other kinds of pattern formation mechanisms. They are all being used by the cell. I'm sure cells is using whatever is possible after billions of years of evolution. But it's another one which seems to be quite rich and explains a lot. I hope these movies run. Well, some of them. Okay, thank you. Maybe it's just too heavy. The movie is just too heavy, yeah.
We already have a question online, so I'll just read that out. Is there, so this is for the part where when you spoke about curvotaxis of cells, is there a trade-off between amplitude and wavelength of the sinusoidal surface over which the cells move? And is there a threshold as a function of cell size?
A trade-off for what?
Trade-off between the amplitude and wavelength of the sinusoidal surface. To cross over, I think.
Oh, you mean that the cells
Yeah.
there's this, we haven't tried that. And as I said here, in this work, we were still limited to a topography which is still changing gently enough so that we don't lose the polarity. We now implemented this robust polarity, which allows us even to reach a wall, lose polarity, and regain it. It's already in the new code. So we can actually answer such a question like, when is the topography too tough to continue along one direction versus the other direction? Yeah.
Is there a threshold as a function of the cell size?
So the threshold that we showed in this work is the threshold of transitioning from preferring to move along the groove to moving perpendicular to the pattern. So that was one nice transition. And it roughly goes where you transition from snugly fitting within one sign of the curvature inside the groove to spilling enough over the edges of the groove to feel the beginning of the ridge. And that already, if your leading edge is there, has such a strong pull to make you, like in the fiber, go circumferentially and go over the fiber. So it's a smooth transition. It's not like a very sharp transition.
Really nice stuff, especially this mixture in the bottom left here. I was wondering how you get more than one phylopodium, because that's what you do in cells. But in your case, it all coalesces into one. But then you were starting to talk about the segregated domains of yellow and red proteins. So could that explain how you get stable but separate phylopodia?
You mean when I mix the red and the yellow together? So, well, you saw that in this calculation, like the one here at the bottom, we got only one phylopodium, and the rest was still happy in a lamellipodia-like structure. And I guess if we load the system with more yellow proteins, or the phylopodia producing nucleators, we'll get more such protrusions. Yeah, now here what's actually interesting is that what is seen here is a case where red and yellow bind equally to each other. So they don't want to segregate, but still, because of the way the yellow form the finger, because more of them are there and they are happy with it, and the red are frustrated, that's how you get this segregation. but the curvature alone is equal in both of them. Yeah.
Okay, I think I'll just move to another online question because there are a lot of them online. Some of the membrane lipids can have a higher affinity for higher or lower curvature regions. I guess this could change locally the bending rigidity. Have you played at all with the feedback between bending rigidity and locally curved protein concentration?
No, we haven't. It's also a question of, yeah, I mean, the short answer is that we haven't here. Wherever you have a curved protein on the membrane, the membrane tries to satisfy the curved protein and get this highly curved shape. But we haven't had a local modulation of the bending modulus. No, we haven't played with that.
Is that okay? This is online, you can't hear.
I guess it's just because, well, I'm sure I know the answer to the question, but I will just ask anyway. In the sorry, as in, it's probably a stupid question is what I mean.
Okay, all of this.
So when you have like
Fine.
the rotating cells on the fibers,
Yeah.
it's not a chiral effect, is it? Like they go either direction.
Yeah.
Just
yeah.
depending, and experimentally, it's also not
Yeah,
chiral.
yeah.
Okay, I was just because I think it was just one example. And I was
Yeah,
like,
there are chiral behaviors in cells, and people attribute that to the chirality of the actin filaments, and because they are chiral, not just the protein but also the filament itself. No, in these experiments, as far as I asked, it's equally likely to go left hand or right hand. Yeah.
Here's another question online. How does the finger-like diffusion behave in a motility simulation?
So we haven't explored that significantly. As you can see from the way it is jumping around here, since it gains some adhesion energy by touching the surface, it does tend to stay in contact with the surface. I think Phylopodia during motility are more involved, if I'm not mistaken, and correct me here if somebody knows, are more involved in it being extended and then retracted. And during the retraction, that they apply a traction force. We don't have contractility here yet, so I guess we cannot describe correctly a Phylopodia-based motility using this model as it is now.
Okay.
Thanks. So, as you said, in the models, you're completely ignoring the internal structure. So, do you think that the fact that your models still seem to kind of replicate some behavior pretty well, that tells us something about the internal dynamics, like that they're kind of stochastic or fast enough that?
So it certainly means that whenever you have these very dynamic cellular structures, like if you see at the top this phagocytosis event or this cell migration where you're constantly moving the cell and changing its shape, or the coiling, all of these are involving highly dynamic membranes. But if you are looking at a very static cell which is adhered to a surface or adhered to other cells and produces a very static structure, usually these structures involve lots of contractile forces, and then they produce a very different organization of the actin, which is based on stress fibers, either along the entire surface of the cell substrate contact area or along the cell contact areas if it's with neighboring cells. So a very different kind of actin, very static, very based on contractility, and that allows adherent and static cells to conform and get their shapes. So I think this model, because it doesn't describe all of these internal structures, I think is more useful for describing cases where the actin is continuously polymerizing and depolymerizing in a very dynamic fashion and producing very dynamic shape changes in the cell, like a protrusion, engulfment, cell migration, etc.
Okay, we have another question online. you showed how the cells move on a fiber, but what happens when they move inside a tube?
Yeah, okay, so that's good. It might, it's not here. So when it's inside the tube, it's a little bit like when it's moving inside the groove, only that now it's an endless groove. You can't get out of this groove because it's wherever you go, you're still inside the groove. What happens is that first of all, it tends to go along the axis, but also very slowly. And often we found that it actually loses its polarization spontaneously and either becomes like happy to crawl along the sides and bridge the tube from side to side if the tube is small enough. And this was observed also in experiments. So in general, they still go along the axis, like inside the groove, but depending on the size of the cell within the tube, it can actually, because it now enjoys adhesion from all directions, lose its polarization completely and be happy just spreading randomly in all directions and lose motility completely. As I said, in the simulations at the moment that we have implemented, we didn't have a way for the cell to regain polarity. But in experiments, of course, you see that after such an episode, it can spontaneously regain polarity. And again, continue to move mostly along the axis, like in an infinite groove. Yeah.
I think we might have time for one last question if anyone has any. Okay, if not, we should thank Nir for the really excellent talk today. And for those who are not aware, we have a couple more talks this series, and do sign up to our mailing list for reminders.